recent molecular advances have proposed a unified genetic hypothesis for FSHD types 1 and 2 and have identified some potential therapeutic targets for clinical trials. This has brought a lot of excitement to the field for individual researchers, but also drug companies thinking about developing new drugs for FSHD. Hand in hand with the advancements that they're making in the clinic comes the need for us as an FSH research community to develop the tools, the outcome measures that we're going to use to measure a response to therapy in the setting of a clinical trial. So this is what I want to talk to you a little bit about today. What are outcome measures? How do we match them to the stage of drug development that we're at? And what are some of the outcome measures that we've used in FSHD? So an outcome measure is a tool that can measure any aspect of the disease. It can measure something in your blood. It can measure some aspect about your muscle. It can measure questions about your quality of life or how you're doing. But I think the key word here is a measurement. It's some sort of number, some sort of metric that we can follow to determine if time A is different than time B, if some essential change has happened in the disease process. It's for use in clinical trials, but we can also use these in clinic. Ideally, outcome measures should be pertinent to FSHD. So we'd like it to be measuring something that's either important to the patient or that clinicians have identified as being important to the pathological mechanism of the disease. We'd like outcome measures to be reliable. So if the disease isn't changing, we'd like what we measure to be the same from one day to the next. But we also want these measures to be sensitive. So if the disease is changing, we'd like the outcome measure to be able to detect that change in a proportionate fashion. We'd like them to be inexpensive. This gets to the feasibility of running clinical trials. If your outcome measures are outrageously expensive, it becomes very difficult practically to make a clinical trial happen. And then we'd like them to be collected at multiple sites in the same way. This standardization of protocols and the ability to collect similar information at different sites is really key to building the infrastructure to run clinical trials. So the right tool for the right job. So we'd like our outcome measures to measure some aspect of a disease that we're interested in following. For example, measuring gene expression's not really going to tell us a lot about whether someone's experiencing pain. We need different tools that will be important at different times in the drug development process. So we're really not talking about one outcome measure that's going to be the be-all and end-all for FSHD. We're really talking about a whole set of different outcome measures that will have their role at different points in the drug development process. So matching the outcome measure to the study aim. So this has a little bit to do with where you are in the drug development process. So when you're doing early studies, these are typically short studies in smaller numbers of patients, and they're often looking at questions like safety and dosing. For these studies, biomarkers become very important as proof of concept or early markers that your drug's doing what it's supposed to do. Things like strength measurements or functional outcome measures are important as you get into this question of effectiveness. These sort of mid-stage studies, they're typically longer in duration. They often require more people. And these are really the go, no-go studies. The studies where we say, do we have enough information here to think we're ready to go on to the large studies to get approval to market a drug in the United States from the FDA or in the international communities from the respective regulatory bodies. And then patient-reported and quality of life outcome measures. These become increasingly important as a drug moves through the process, and they really judge the overall impact of a treatment on both the physical and the mental aspects of a disease. So biomarkers, as the name implies, it's something that you measure in blood or tissue. It can be gene expression, it can be protein levels, Other measurements like neuroimaging, and I'll get into this a little bit, can also serve indirectly as a biomarker. They're important for these early studies, where small numbers of people, where you really want an initial signal that a drug's doing what you think it's going to do, and also for proof of concept. If the idea is that a drug is going to turn off protein A, you'd like to be able to measure protein A and show that it's being turned off. Ideally, we'd like a change in these biomarkers to predict some change that occurs later on in the course of the disease that's important to people. For example, predicting whether or not you'll need to use a cane or a walker at a future point in time. But they don't have to have that tie in order to still be useful. So what are the biomarkers that we have for FSHD? Well, DUX4, the protein you heard a lot about, I think, yesterday, well, this might be an ideal biomarker, but it's very difficult to measure directly in patient tissues. The background to the slide is an image from the Tapscott lab, and I like it a lot because it shows a string of nuclei from a muscle culture in the background, and that pink color is DUX4 being expressed. But short of measuring DUX4, other genes affected by DUX4 or downstream from it may be more persistent, and these may actually serve as really valuable biomarkers for early studies. Other proteins in your blood or serum may be also important, either related to the disease mechanism or indirectly related. Proteins that may show you indications of muscle turnover or oxidative stress. However, more work's needed to determine which biomarkers are going to be important in FSHD, and it may be that different biomarkers will be important for different types of drugs. So it may be partially determined on what kind of drug you're measuring. MRI may serve as a novel, non-invasive biomarker of disease progression. MRI uses magnetic fields and radio waves to take advantage of certain quantum properties of water molecules to get us really sophisticated information about the structure of muscles. There's been some indication in FSHD that changes on MRI may give us some signs of disease activity in FSHD. And an ability to target some muscles that are at risk for progression. The picture that you see here on the screen is from an Italian study, and under the label T1, what you see is the arrow pointing at a muscle that really essentially looks normal on that MRI sequence. Underneath the T2 stir label, what you see is that same muscle shows up bright white. And in this Italian study, they biopsied these bright white muscles, and what they found was inflammation. In a certain number of the muscles, they found some molecular markers that there was disease activity related to FSHD in these muscles. So this MRI stir sequence may give us a way to identify muscles that are at risk for progressing, and also a way to monitor a response to therapy. Another aspect of MRI that might be very useful in clinical trials is quantitative measurement of the fat in the muscle. This is more and more becoming used in different types of muscle diseases. We see it being used in the limb-girdle muscular dystrophies and Duchenne muscular dystrophy. And what I'm showing you here on the right in that figure is some data taken from a study that was completed in the Netherlands that I think you may hear a little more about later. But what it shows is on the y-axis is the ratio of fat inside a given muscle. Each of those little circles is an individual muscle that was measured. And what you can see in that graph is along the bottom, there's a whole lot of muscles that are essentially normal. They have a very low or almost no fat content in them. On the top of the graph, what you see is there's a lot of muscles that are really almost completely replaced by fat. So these would be muscles that are already, very weak, far along in the process. But what was most interesting was this intermediate group of muscles, these muscles with an intermediate fat content, because what they found is they were most likely to progress to change over a very short period of time of 3 months. So that means measuring the quantitative fat may be a very effective way to get an early signal that a drug is doing something in a muscle that might be useful to follow. So the workhorse for clinical trials in muscular dystrophy has really been measuring strength. Strength would intuitively seem very meaningful in a disease that's primarily affecting the muscle. There are two very well-studied methods for measuring strength in FSHD. One of these is manual muscle testing. The other is quantitative myometry. And many of you in here will have had one or the other done on you at one point in time. Both of these techniques test strength against fixed resistance or isometric contractions of the muscle. Quantitative myometry uses a digital force transducer. This is the quantitative part. And what you can see in this picture is an example of the common apparatus for this, which is a table that has a large metal frame around it. And the force transducer is connected to an inelastic strap, and a person's put in a position to isolate the strength of a single muscle. So there's standardized positions for doing this. You can also use handheld strength gauges to do this. Manual muscle testing, on the other hand, tests your ability to pull or push against an evaluator who's been trained in standardized positions and procedures and has been trained in using a standard strength scale that ranges really from normal strength to no strength at all, where somewhere in the middle is the ability to contract that muscle against gravity but not against resistance. So both of these methods have proven to be reliable. And you can average muscles across multiple muscle groups to get a combined score that you can think of as the average overall strength of an individual, which you can follow over time. The data in this graph is from one of the longest natural history studies in FSHD, and it shows you a change in strength on the y-axis, months of follow-up on the x-axis. People came in every 6 months. Each of the plus signs is an individual. The dotted line at 0 would be the change in strength compared to the first visit. So if you stayed at that 0 level, that would mean your strength wasn't changing at all. And what I really want you to look at is that second group there, because that would be 1 year of follow-up. And there's a couple things I want to point out. First of all, the length of that line, it's quite long. So there's a lot of variability from individual to individual in the changes in strength. There's some individuals above the dotted line. These would be people who got stronger over the course of a year. And there's individuals below the line. These are people who would get weaker over the course of the year. The X is the average change in strength. And so what we saw is, as a group, people lost about 3 to 5% of their strength over that year. However, small changes in strength can be very difficult to put into a clinical context. What does it mean to lose 3% of your strength? And that gets to this idea of functional outcome measures. These are things like the time to go up some stairs or to walk a given distance or to lift objects over the head. The FDA is really interested in these outcomes because intuitively it seems like activities that correlate to things that you do in everyday life would be meaningful. In FSHD, we've looked at a number of these, and they show moderate to strong relationships to disease severity or measures of strength. But when you look at populations of individuals, we don't really see them changing over time. And this is time periods up to 3 years of following people. That doesn't mean that for a single individual they wouldn't have had a significant change in one of these tasks, but in groups of individuals it's very hard to document a change. Patient-reported outcomes can be standardized questionnaires. Some of these aren't disease-specific at all. Some of you may have heard of the SF-36. It's probably the most common quality of life outcome measure used in clinical trials. The SF stands for short form, the 36 for the number of questions. It asks you questions about your physical health and the impact of your physical condition on your mental health. The Promise 57 is a version of this kind of a form that was developed by the NIH and has the distinct advantage of being freely available. The INQUAL is a little different because it's a little more specific to neuromuscular diseases. All of these scales have been validated and are really ready to go for clinical trials. There is one scale that's being developed by a collaborator of mine at the University of Rochester that's an FSHD-specific health inventory, And the way he did this was he started with patient interviews and determined domains or areas that were important to patients to develop the scale. And in the box, you can see some of these areas that range from problems with the shoulders, with mobility and walking, but also things like fatigue and pain and the change in body image due to the disease. Now, I'm running a little short on time, so I may skip this slide or just go over it briefly. There are a lot of outcomes in development. In fact, as many researchers as there are who have an idea about what would be good to measure in FSHD, there may be that many outcomes, but at a certain point, it's important that the procedures and the equipment for measuring these outcomes become standardized. The three that I've put on this slide, I think, are reaching that point where they're getting close to being able to be used in development. And these are electrical impedance myography, and I want to mention it here because the FSH Society has been kind enough to support me in collecting data on this outcome. Reachable workspace, which uses a very novel 3D camera system that you can get with the Xbox Connect to discover how much space someone can reach with their shoulders. And MRIS, this is a different aspect of magnetic resonance imaging where instead of looking at the structure, they're really looking at metabolites in muscles. If anyone has questions about these, please feel free to ask me after the talk. So how many people are enough? This really depends on the outcomes variability, the variability across individuals, and the variability within a given individual, and how big of effect. The smaller the variability or the larger effect, the fewer number of people that will be required to do this. A better understanding of our outcomes can help us plan trials more efficiently. And I just want to conclude on this notion about coordination between sites. In the end, we're a community and we're going to come up with these outcomes together. The ability to standardize protocols so all of us who are looking at the same thing are collecting them in the same fashion will help us build a network of sites that are ready to run the clinical trials. So we'll be ready to go. Thank you very much for your time.